Well, today's lecture covers the last part of the protein, and in here we're going to discuss more about the protein folding mechanisms as well as the protein cellular functions. The proteins can be folded in either native or non-native states, and in theory, any peptides could adopt many different secondary or tertiary structures such as alpha helix or beta sheets, and also different kinds of domains depending on their amino acid sequences within the polypeptide. And remember that each amino acids have 6 torsion angles if you include the side chain that would be 7. And depending on the combination of those, you can actually get a lot of different secondary structures of the polypeptide. But in general, actually, all molecules, the protein molecules of a given protein species adopt the same, just one kind, 3D conformation within the cell. And this structure is called the native state of the protein, where the protein has its designed, its own cellular function, as opposed to non-native state of the protein, will basically folded in different structures other than this native state, which will lose, in most cases, lose its own function and can actually obtain some other function that can have an implication to a human disease. So the native state is usually, but not always, the most stable state or the form of the folded protein. So that means it has the lowest energy within that protein. And disruption of the native structure is called denaturation of the protein, and that can be happened by the breaking the weak bonds as well as some strong bonds too, responsible for secondary and tertiary structures. And weak bonds include the hydrogen bonding as well as van der Waals forces. And the results is a protein non-native, again, the form or the state in that the protein loses its own function or denatured the state. And denaturation can be caused by raising or rarely lowering the temperature. An example is a boiled egg. Obviously, it loses its own function or the structure and even the color too. And extremes of a pH is another causation for the denaturation. And chaeotropes is also can cause the denaturation. And examples of the chaeotropes would be the guanidine hydrochloride or the guanidine ion, as well as 8 molar, which is a really high concentration urea that can disrupt the interactions within the protein. And also detergents does cause the denaturation too. And most denatured proteins tend to precipitate because they expose some regions that is not soluble, meaning that it's non-polar regions. So they tend to interact with each other to get the high like a volume of aggregation of those such proteins to result in the precipitation. Denaturation and renaturation, which means that it recovers the form from the denatured form of a small protein RNase A, which is a pancreatic ribonuclease, has been shown in a famous Anfinson's experiment in 1957, which earned him a Nobel Prize. And what he did basically was that he started with the RNase A that is in a native state, which is an active enzyme, and then he treated with the 8 molar urea as well as mercaptoethanol to break the disulfide bonds. So it's denatured form here, and then he removed the both urea and the mercaptoethanol and observed that it can be recovered as just a native form, which is an active enzyme. So this process is called denaturation and renaturation. What it did also was that instead of removing both of them at the same time, he just removed the mercaptoethanol only to recover the disulfide bond. In this case, wasn't sufficient to forming the renatured into the active form or the native state of the protein there, but by adding the a little bit more of the mercaptoethanol on this scrambled or the inactive form, he could actually recover the function or the activity of that enzyme, which tells us that this amino acid sequence itself in the polypeptide seems to be sufficient to specify its 3D conformation. So whatever the treatment that you have done, at the end with a little bit of tweak, you can actually get the recover the whole functional protein structure here. And it is also called Anfinson's dogma, where the basically protein folding is a spontaneous process that does not require the assistance of extraneous factors, but it can just fold it in itself by itself. Let's say that you're doing a thought experiment where you're watching protein folding, and since protein or polypeptide has several amino acids, if not many, each amino acid have different torsion angles, so different conformations can be exerted from that too, and also side chains can be oriented in different angles. So there are so many different kinds of combinations of the conformation that you can get, and for that protein to find the best conformation that has the lowest energy, it might take some time to fold it properly there, but when scientists observed the actual protein folding events, they found that that event happens within seconds or even less, both in vitro system as well as in vivo system. And as you know, in vitro is basically out of the cellular system, so it's going to be on the dish or in the test tube, as opposed to in vivo system will be some events or the experiments that is happening in the cellular system or the living organisms. The protein folding is so fast and much faster than what scientists expected, and that is basically the Leventhal paradox, and that happens in both in vitro as well as in vivo, far too fast for it to be a random searching process for the best conformation. But the RNA fold is even much faster in vivo than in vitro, meaning that now some kind of in vivo components can be involved in the folding too. Now let's talk more about the Leventhal paradox, where it shows the difference between a thought experiment, like I explained, with all the amino acid residues finding their best conformation by themselves, which can take time, and the actual protein folding in vitro and in vivo, which takes much shorter time. So let's consider a protein with the 100 amino acids, and let's use a very simplified model here where there are only 3 possible orientations per residues, 2 torsion angles which are phi and psi. So this again still ignores the side chains, which can generate a lot of more possibility for the conformations. So since we're ignoring that, it's going to be a huge underestimation. Anyway, that would be the 3, which is the 3 possible orientations, with the power of 2 for the 2 torsion angles, to the power of 100 for the 100 amino acids. So it's going to be 2.7 times to the 10 to the 95th power of a different conformations in total, which is a huge number, and assume that one conformation can form every 10 to the minus 13 seconds, so that's 100 picoseconds. Let's say that it takes really short time with that, but still, if you multiply this all different conformations with the time spans for each conformation, it's gonna be more than 8 times 10 to the 74 years to correct the fold of protein. It's the maximum value. So let's say that the average is the midpoint. It'll be still a really long time to find the best protein folding conformation. So obviously, not all conformations can be sampled during the folding because it takes much shorter time. So there could be different mechanisms or different rule that is applied to the polypeptides to form the proper form. So the new folding view is needed for that. Well, that Leventhal paradox was basically based on the random process where each of the amino acids or the multiple amino acid residues find their best conformation to find the lowest energy point. But life, the reality, and the protein folding is not a random process. Now let's resolve that paradox based on the knowledge that we gained after this paradox. The first one is that there are a limited number of secondary structure elements. So rather than all amino acid residues form all different kinds of conformations, they can form the either alpha helix or the beta sheet or the turns or the loops and things like that. So they have some rules for their secondary structure. So there are going to be some limitations or the smaller number of possibility for the conformation within that small region. And these elements tend to form spontaneously during the co-translational folding of a protein, which means that it's not going to be the whole thread or the whole polypeptide that is generated and then the folding happens. It's more like instantaneous and spontaneous. So whenever there's a nascent polypeptide or the little threads that is generated from the mRNA through the translation by the ribosome, it'll start forming the secondary structure right away. So it'll save a lot of time too. And proteins fold via so-called folding landscapes, meaning that it has different steps or different processes that can fold first, and then the next step it can fold the other levels to form the whole like a 3D protein structure. So this landscape is the protein follows the pathways or the rules of a folding that leads to the correct three-dimensional structures. And this there are several rules for that. And the disulfide bond bridge is one of the rules that you can actually follow as the pathway or the landscape here too. And folding intermediates may be important in such folding landscapes and pathways. So there are always intermediate stages where small regions can be folded first, following the pathway or the landscape. So instead of looking at the whole complicated 3D protein structures or the amino acid residues, a simplified view for explaining this protein folding would be looking at a folding funnel, which indicates the energy or the internal energy that molecule has. So it starts with the top here and it collapses down to find the lowest energy point. But of course, the entropy would be much higher on these higher regions. But as you can remember in the thermodynamics, the Gibbs free energy or the driving force for spontaneous reaction to this finding native state will be the combination of internal energy or the work and the entropy. So by sacrificing a little bit of entropy or disorderly, you can actually gain a lot in terms of decreasing the energy to find the most stable condition when you look at this folding funnel. So like I told you, there are only limited number of secondary structure elements such as alpha helices, beta sheets, and turns, meaning that instead of you have to take it into account of all different pathways or the landscape here, you can actually follow some of the probable ones which can form these secondary structures. And folding can be thought to occur along energy surfaces or the landscape here on the funnel. So when you look at this energy landscape, basically the folding process itself is to descend towards the free energy minimum state, which is the native state or the most stable conformation. And the A and B pathways are shown here, which A goes to the smooth surface of this energy landscape. So it's going to be just rapid folding, as opposed to the B pathway has a little like bumps here, meaning that it has a secondary energy minima here, so it can be actually trapped here a little bit and needs some more help or the energy to overcome this secondary minima to get to the actual native state. Just like the protein gliding through the energy landscape to find the energy minima, proteins fold in stages, meaning that the proteins can have local folding through nucleation of small clusters of residues. So here are the general order of folding. So first one is short regions rapidly form small stable secondary elements, which takes about 5 milliseconds or so. So it forms these beta sheets or alpha helices and things like that. And after that, there's a small secondary structure elements interact with the other local elements to fold into globular units, which typically forms the hydrophobic cores. And it can also generate the super secondary structure on an intermediate time scale. And associations are thought through the various weak chemical interactions, such as van der Waals interactions or hydrogen bondings. And it typically takes 5 to 1,000 milliseconds or so. So still pretty quickly that those process happens. And these globular domains may be a complete small protein, just like the RNAs or the lysozyme, the small proteins, or a number of these in large proteins can interact more slowly to fold into the final folded structure. So it's going to be huge protein with the multiple domains. And the last step will be the assembly into the multi-subunit proteins. So multi-polypeptide form the polymers and so on. And it's not all the cases, but for some proteins, definitely can make the higher order structures. Now let's talk more about the co-translational protein folding, where protein folding process and the translation or the synthesis of the protein are happening at the same time. So that's why it's called co-translation and the protein folding. Initially, the first 30 amino acids or so of the polypeptide chain present within the ribosome are constrained and cannot fold until they exit from the ribosome to have the enough space for folding. And as you know, the amino or the N-terminus emerges first and the C-terminus emerges last. So the folding process will be follow the same order. So N-terminal regions will be folded first and then follow the C-terminal regions for the protein folding. And as soon as the first part of the nascent chain is extruded, it will start to fold co-translationally. So both translation and the protein folding happens at the same time, which makes the secondary structure of these N-terminal regions and again continuously to the other regions of the polypeptide. And as the complete polypeptide is produced, it will fold in a similar fashion, and then the final tertiary structure will be established in some cases, like the larger proteins. And also the coordinated structure can be formed after that by assembling the subunits or the multiple polypeptides into a big globular protein structure. Molecular chaperones are basically helper protein for the protein folding, but the great majority of the proteins can fold without assistance by chaperones, but they can do that by themselves in a co-translational manner, although many must refold after transport through ER and Golgi for the other cellular functions. But this is not necessarily true for many big proteins where it has complex structures. So these big proteins may have difficulties reaching their native states by themselves, so must be stabilized by the molecular chaperones or the chaperonins by assisted folding. The mechanism basically for this chaperone working is that it binds to the nascent or emerging polypeptides and stabilize them by associating the hydrophobic residues. Otherwise, if that doesn't happen, these hydrophobic residues on those big proteins tend to associate with the other hydrophobic regions within that big protein, or the inner molecular associations with other proteins that prevent proper folding. So there are dozens of different types of molecular chaperones, and some accomplish functions different from just helping protein folding. So examples are some help protein assembly, some help transport proteins to various parts of the cell, and some help damage the protein refold. There are several classes of chaperones based on their size as well as their functions. These are four major classes. First one is heat shock protein, or HSP70, which can work as a single unit, and also with the HSP40 as co-chaperone. And the second one is HSP90, which has a lot of implication in neurodegenerative diseases, and also is involved in regulation of a protein aggregation. And the third one is trigger factors, which are associated with the ribosome to prevent improper folding during translation. And the last one is chaperonin, which are large multi-subunit proteins. And all these chaperones are ATPases, meaning that they hydrolyze ATP to bind and release unfolded proteins. And this ATP hydrolysis process basically generates the energy, so it powers this expensive process to unfold, to correct the unfolded or misfolded proteins. And the most extensively studied chaperone is the GRO-EL, GRO-ES complex of C. elegans, and the structure is shown here with the side view and the top view here. And it basically consists of 14 GRO-EL rings here, which forms the sort of the huge barrel that can, little misfolded protein that can fit in, and also a cap here, which is a GRO-ES. This reaction cycle shows how GRO-EL, GRO-ES complex in E. coli works to correct the protein folding. So this first structure, which looks like a barrel, comprises of the total 14 GRO-EL subunits, where 7 of them located on the top and the other 7 located on the bottom. Again, there are 2 openings on top and the bottom, and the one that is exposed, especially for binding to misfolded protein, has the ATPs attached to those GRO-EL subunits. So once misfolded protein is introduced into this cavity, then GRO-ES, which works as a cap, placed on the top of this GRO-EL complex. Now it seals in, and that induces the conformational changes. So you can see that it's a little bit expanded here to fit better this misfolded protein. So this ring particular barrel region where the misfolded protein is located is called cis ring of the GRO-EL complex, and the other one that does not have misfolded protein is called the trans ring. So within the cis ring, there's ATP hydrolysis happens, so it changes to ADP, and with that chemical energy, actually that helps this protein to be refolded to its better shape with a lower energy. Once it is properly folded, now another improperly folded protein or misfolded protein can be introduced to the other side of this barrel-like shape. And then that induces again the conformational change of this bottom region, and also induces the release of both the cap, the GRO-ES, as well as the better folded protein. And this process can now, this barrel can flip over like that, and the process goes on. And this better folded protein can actually be introduced again within this complex to be even better folded to become to reach to the native state. Protein misfolding can cause serious human diseases such as prion-based and Creutzfeldt-Jakob disease and the mad cow diseases, and many of them are actually neurodegenerative diseases. And basic mechanism of such diseases are quite similar, as the formation of protein aggregates or the aggregations that can affect or to kill the nerve cells. And prion disease are self-infectious too, which means that this misfolded version of a prion protein, which is basically beta strand based structure, can give a rise from in a very rare case, but once that is formed, then now it can affect the other normal structure of the prion proteins by making the heterodimers or the polymers in there, and affect this normal shape of the prion protein to the infectious shape, which is beta strand based, and it's also called prion protein scrapie version, so PRPSC too. And this now the homodimer of the infectious protein structure can affect all other normal prion proteins to make the amyloid protein aggregation. This image shows the cross beta filament structure resulting from extensive stacking of misfolded beta sheets. So beta sheets, the problem of that is that now it can be really stacked together without too much space, so it can be fully packed there, which can be resistant to any kinds of proteases action, because they are so packed together. There's no little region for the protease to attack, and this shows the model for the conversion from the prion protein, the normal one, to the scrapie prion protein, or the PRPSC, and that shows that two alpha helices here in the normal structure changes into the 4 beta strands here. Again, it has ability to be stacked together really in a packed manner. And the largest protein structure would be the protein quaternary structure, which is an association of multiple polypeptides into a functional unit. And many proteins are engaged in this type of protein-protein interaction and forming the quaternary structure, but not all of them. And individual proteins in the quaternary structure are called subunits. And an example is again the hemoglobin, where it has 2 alpha subunits and the beta subunits. After translation and protein folding, which often happens simultaneously, the protein modifications happen, which are often required for the activity as well as the maturation of the protein. So they are basically cleavage and the covalent modifications of the proteins, and oftentimes they happen after protein synthesis, but also can happen co-translationally too. So the first type of protein modifications that we're going to discuss is cleavage. Some proteins require sections of the polypeptide chain to be removed for correct maturation, and a zymosan is a catalytically inactive protein precursor that is a good example that has go through the series of cleavages to become an active form. So this zymosan must be cleaved proteolytically to be activated. An example of zymosan and the cleavage modification would be the insulin, where it shows the really kind of simplified structure of that protein. So first it is synthesized as a pre-pro-insulin, but with the first cleavage, which removes the signal sequence at the N-terminal region of the pre-pro-insulin, it becomes the pro-insulin, but it's still inactive until there's a second and third cleavages happens, which removes the joining of these 2 polypeptides. But these 2 polypeptides can be joined together by the disulfide bonds to keep it its structure, and also now it becomes an active enzyme. And another type of the protein modifications are covalent modifications, which are hundreds of types known already, and these modifications can be required to obtain the active conformation. And an example for that is a collagen that we're going to discuss on the bottom here. And also these modifications can be used to control the activity of the proteins. And the histones and the signal transducing proteins use these such modifications. Here's an example as a collagen here, especially the proline residue within the collagen have the covalent modification to the hydroxyl proline by adding the hydroxyl group on the proline here by the hydroxylation. And this process requires a vitamin C, meaning that if there's a lack of vitamin C, there's going to be loss of a hydroxyproline, which will lead to the scurvy disease. And due to a weakening of collagen fibers, especially over time, the hydroxylation of the prolines stabilizes the structure. So it is definitely required and have to be maintained. Phosphorylation is another type of protein modifications that is widely used, especially among signal transduction proteins. What they have is a phosphorylation of hydroxyls on specific residues. And these proteins become transiently phosphorylated which leads to the conformational changes in many cases, or also can lead to the changes in the partner proteins that they're interacting with. And that can either activates or inhibits their activity of those phosphorylated proteins. And phosphorylation can be on one of three different amino acids: serine, threonine, and tyrosine. And a particular protein will only have a specific modification sites. So that means not all serine, threonine, and tyrosine will be target for the phosphorylation, but specific ones only. And they can change basically to phosphoforms: phosphoserine, phosphothreonine, and so on. And together, these modulate the protein activity. And there are two different types of protein too, that are involved in phosphorylation: kinases and phosphatases. Kinases are the cellular proteins that phosphorylate these residues here, as opposed to phosphatases are the cellular proteins that remove the phosphate groups from the phosphorylated proteins. The shape and structure of the protein and the domains within the protein make protein function. And these are some examples of the protein functions. The first one is a structural function, where collagen and elastins are included, and they work as a scaffold or the building blocks. And mobility is another protein function, and actin, myosin, and tubulin and flagella are the examples. And receptors and ligands are involved in cell signaling pathways, and insulin receptors and insulins are examples for that. And protein also work for the immune system, and example is an antibody that detects the antigens. And housekeeping function too by the chaperones, which correct the misfolded proteins, and also involved in signaling as a biological function. And kinases and proteases are the good examples for that. And also they have enzymatic functions, so they catalyze some biochemical reactions, and proteases are the examples for that. And also protein works for the storage and transport, and hemoglobin is a good example for that. So among these different protein biological functions, in this lecture we're going to focus on this storage and transport functions by discussing more about the mechanism of how hemoglobin works. The globins have a critical role in oxygen transport and storage within an organism, and there are majorly two different types. One is myoglobin and the other is hemoglobin. They have a similar shape here, as you can see, when you focus on the one subunit of hemoglobin and compare that to the myoglobin too. But of course, hemoglobin have four subunits, two alpha and two beta here, which form the quaternary structure here. And hemoglobin located in the arteries and the veins, basically involved in the respiration as well as the circulation too, as opposed to myoglobin is located within the cell for the cellular respiration. Myoglobins are involved in the storage of the oxygen molecules, as opposed to hemoglobins are involved in transport of oxygen. Then how to transfer the oxygen molecules from lung to cell so the cells can get enough oxygen for their respiration. The living organisms use the partial pressure of oxygens and regulate that, which is indicated as PO2. And as you've learned in the chemistry or the organic chemistry classes, the partial pressure of a gas molecule is basically the amount or the concentration of that in the solution. So the hemoglobin need to pick up the oxygen molecules in the lung, which has the high PO2. And this hemoglobin releases the oxygen in capillaries, which have moderate to low PO2. And the cells within the tissue have even lower PO2, so it can easily get the oxygen from the capillaries too. And by the myoglobin, it can store the oxygens until required. And P50 is the term that is used to indicate the PO2, where 50% of the heme sites are filled, meaning that 50% of heme have the O2 bound. One major difference between hemoglobin and myoglobin is the affinity for oxygen. So hemoglobin has moderate affinity, as opposed to myoglobin has a high affinity. So that is basically optimized for their functions. Myoglobin works for the storage, has the high affinity to get the oxygens into the tissues and the cells, as opposed to hemoglobin has to bind the oxygen in the lung, but have to release it in the capillaries. So it has to have a moderate, so it can easily control the affinity too. And during the pregnancy, the fetus must get the oxygen from the mother's bloodstream. So this fetus expresses a special fetal hemoglobin that binds the oxygen molecules with a higher affinity than the adult hemoglobin. So here are more differences between myoglobin and hemoglobin. Like I discussed earlier, they have different quaternary structures. Myoglobin is monomer, but the hemoglobin is tetramer, and they have different affinity for oxygens. And also this myoglobin, its function is unaffected by the pH, carbon dioxide, nor bisphosphoglycerate or the BPG, but the hemoglobin is pretty sensitive to those factors. And also myoglobin binds only one oxygen molecule, as opposed to hemoglobin binds four of them because of the four subunits. Myoglobin consists of 153 amino acids, so it's relatively small, and 77% of it forms the alpha helix. So it contains 8 helices in total from A to H, and interior residues are nonpolar except for the couple of histidines. But exterior residues include both polar and nonpolar amino acids. And importantly, it contains a heme cofactor, which is located here in the middle of the myoglobin. It forms it is with the porphyrin ring and the iron divalent ion, and required for the protein folding of that myoglobin, as well as critically the oxygen binding too. So oxygen binds specifically, especially to this iron divalent ion. So the heme is a critical functional group in the globins that binds the oxygen reversibly. But gases like carbon monoxide also bind to the heme, but even more strongly than oxygen molecules. And that explains why these molecules are highly toxic. Then what role does the protein portion, the globin portion, except for the heme group of the myoglobin play? It actually discourages the binding of carbon monoxide and other toxic gases. How it does is that this other protein portion forms the pocket, which fits the best with the oxygen molecule. But even if the carbon monoxide have an even stronger affinity to the heme, the pocket itself does not fit well, so it will actually discourage from this carbon monoxide binding to the heme group and be stabilized. And it's not just the heme itself that binding the oxygen molecule, but it's coordination with the other protein residues within the globin to intensify and optimize this binding. So these are the specific residues that are involved in oxygen binding of the heme. This plot shows the oxygen binding curve for myoglobin, where x-axis indicates free oxygen that is expressed as the partial pressure of O2 or PO2, and y-axis shows the YO2, which is a fractional saturation of the myoglobin by oxygens. And as you can see, this shows the hyperbolic curve, so there's a dramatic changes at the beginning here where x values are small, but now when as the x-axis values gets bigger, it has a smaller changes on this YO2. That means when there's a little free oxygen present, there are gonna be the dramatic changes in terms of the saturation of the myoglobin. So myoglobin will quickly pick up the oxygens and will be saturated. But as there's a higher amount of oxygens there, myoglobin will not change in terms of its fraction for the saturated portion of with the oxygen. And also, when you look at the P50, which is the midpoint for this fraction of the saturated myoglobin, that has a quite small number for the free oxygen as 2.5 or so. And this low P50 value means that there's a high affinity of this myoglobin to the oxygen. if this curve were to be more like a flat, like this, then that would mean that it'll be more like a gradual changes depending on the free oxygen. So the P50 will be much larger for the in terms of this PO2 values, and that means this will have a low affinity to that oxygen. So far we talked about the myoglobin. Now let's talk about the hemoglobin, which has four subunits, two alpha and two beta subunits. They are in similar size as 140-ish, and they form the tetramers. And each alpha and beta subunit individually looks like a myoglobin, but when you actually look at the sequences of them, only 24 amino acids are conserved among myoglobin and the alpha and beta subunits of the hemoglobin. But there are many different ways to get the same foldings, not just the sequence identity, because normally it needs just 15 to 25% of the amino acid identity to become the protein with the same foldings or the same shapes. So they can still, the hemoglobin and myoglobin still form the same, almost identical folding there and the shape there. But the main differences between the hemoglobin and myoglobin are due to the quaternary structure of the hemoglobin. So hemoglobin has a cooperative binding of oxygen between these subunits, and there's a allosteric regulation by the carbon dioxide, hydrogen ion, or the pH, as well as the BPG and the DPG. A special binding curve is required for an oxygen transport protein hemoglobin because it has to have a high affinity at a high oxygen for the effective loading, but also it has to have a low affinity now at the low oxygen for effective unloading too on the tissues and the cells. So when you look at the binding curve of the oxygen, here it's the same format as the PO2 or the free oxygen is indicated on the x-axis, and MyO2 or the saturation, the fractional saturation of the hemoglobin is indicated on the y-axis. And you can actually compare those curves between myoglobin, which has a really fast curve, and the small P50 value, to the hemoglobin curve, which is more flattened here. And hemoglobin has basically this kind of S shape, the sigmoidal O2 binding curves. And this type of curve is very efficient since it permits the full saturation of the protein, hemoglobin, in the lungs or the gills where it has the high oxygen, but with just the subtle changes in the free oxygen concentrations can result in dramatic changes in terms of the saturation of the hemoglobin now. So it moves to the tissues and the cells. Now it can effectively release the O2 because of this low YO2 values. And this is a special feature of the hemoglobin which shows the sigmoidal oxygen binding curve is due to the cooperative oxygen binding. And such a binding curve reflects the weak versus the strong binding at low and high oxygen pressures respectively. And this is due to the existence of a cooperative or allosteric interaction among the oxygen binding sites, because they have four of them, four heme within the hemoglobin molecule. In other words, the filling of the first site of the heme increases in the affinity of the other sites for the oxygens of the other hemes in the other subunits. And this phenomenon requires communication between the different subunits regarding their oxygenation states, either filled or empty. But either one of them happens, then it affects the other by their conformational change, and there's a communication between them to sequentially or the cooperatively change all of those subunits within the hemoglobin. And it is only possible because of the quaternary structure of the protein. So myoglobin cannot achieve that because it has only one molecule or the polypeptide. It only happens between the four subunits working cooperatively. So again, this is a transition point from the weak to strong binding in the middle of the sigmoidal curve. but the weak point, there's the weak binding happens when the oxygen is low, and the strong binding happens when in the presence of a high oxygen pressure. So like I briefly mentioned in the previous slide, significant changes in the overall quaternary structure of hemoglobin can be observed upon oxygen binding. Especially there's a rotation and sliding happens from deoxyhemoglobin to oxyhemoglobin. So it brings the beta chains closer together if you compare that to here. And also there's angle changes in terms of the quaternary structure itself, and also narrows the central cavity. And oxy state has the higher oxygen affinity now, and the switch to this state is what causes the cooperativity in the binding of oxygen. The heme causes the structural change of hemoglobin through the helix F. Binding of oxygen of the heme group causes the pull of helix F downward, resulting in the conformational changes throughout the protein too. So the blue structure here is shown when the heme is not bound to the oxygen, which is a T state or the tense state, as opposed to the red structure that is shown here is the R state or the relaxed state in terms of this heme, where the heme is bound to the oxygen molecule. And as you can see, this helix F is downward, so it's pulled down here. So there's going to be a conformational change. When this histidine, the critical residue for interacting the helix F and heme, is mutated to the glycine, still there's another free imidazole that is can be used to bind the heme, and this mutant hemoglobin still binds the oxygen because the heme group is unaffected, but it will not show any cooperativity because it loses this linkage here. There are other factors affecting the oxygen affinity of hemoglobin. The first one is the 3-bisphosphoglycerate or the BPG, which lowers the oxygen affinity of the hemoglobin, but not the myoglobin. So it affects the cooperativity of the hemoglobin. And second one is the pH, especially the reduction of the pH lowers the oxygen affinity of hemoglobin by the Bohr effect. And the third one is release of the carbon dioxide or its accumulation in the respiring tissues, which lowers the oxygen affinity of the hemoglobin too. And a disease connected with the hemoglobin is sickle cell anemia, which is resulted from a single mutation in the beta chain from glutamic acid to valine. And minus charge in the glutamic acid is changed to a nonpolar hydrophobic group, valine. And this site of mutation is at the surface of the protein, the deoxy form of the hemoglobin. So it results in the beta chains stacking together in the deoxy form. So there can have this accumulation of this hemoglobin and sticking together. So it inhibits from the binding to the oxygen molecules, as well as it inhibits any kinds of conformational change too easily within this accumulation.